Okay, here we go. And it's our last class meeting. And and we're making progress toward our final exam, and you're making progress on your capstone project. Great. And let's just make sure we get this going here. Okay, fabulous. All right, so no more classroom discussions, of course, no more reading assignments, except you'll see an optional one down there. So working on your capstone project paper and journal, and we talked a lot about that. We can still meet about it, and send me an email when you're done with your work. Then we have our final exam a week from today. It starts at this time in this classroom, and then we started talking some about the role of ecological developmental biology and evolution on Monday. And we'll keep talking about it some today. And one of the exam questions is going to be about it, but you're going to have options. So you could choose to not write about that topic. But as talking about it, it's a very interesting topic, and we just don't have time to keep going into it in detail. But it popped up a couple of times in our course, in our student discussions, and so I'll talk about it more today. And I realized I should give you the option of some reading, so I'll put some optional, totally optional reading that would be if you're interested on a topic, or if you decide you want to write your question, write an answer about that question on the exam. Okay, that makes sense? And so, and then if you don't have time, well, you could download the reading and read it, this summer at the beach. Okay, so that's the flow. And then remember on the final exam, we're trying to prepare so you can use different examples on the different questions. And we're going to look at the ideas about the questions. We talked about them some on Monday, and we're going to look at them again in a minute. But this idea is that you should not have the attitude of you're going to prepare one universal example, like social insects, workers, and queens. And it's a perfect example of all the topics in our course. Instead of doing that, we've talked about a lot of different examples, and you should be ready to plug in a lot of different examples and describe them some in detail. And remember, we're always interested in ranging on our examples from the ecological context that they're in. What is the story? What's happening here? Whether it's the first half of our course or the second half of our course. What's the situation up at that level? And then what's essentially the initiating factor or the relevance, and then we dive into the cellular and molecular mechanisms, tissue mechanisms like that in the organism, because they're great principles we've run into over and over in our course there. And so you want to be prepared to have that, and then ultimately, what impact does this have on the phenotype of the organism, whether it's the first half, natural phenotypic plasticity, or the second half, disruption of it. Okay, so strive for that in your preparation. Okay, so then as you're preparing, this is a good thing we talked about during our course to try to organize your learning, and you can organize your planning for exams and exam answers like that as well. Remember on the first exam, I said, here are the questions a couple days before the exam. This one, we're talking through possible exam questions, and I said, you're gonna have to write 5 20-point answers, and here's some possible questions. And I also suggested that if you had a question that, a suggested question for the exam, you could send it to me. And you could still do that, but we have to have that very soon because I have to share it with everyone, not just you, and say, yeah, okay, Luke, I'll put that on the exam. That'll be a good one, like that. I need to share it equally with everybody so they can say, oh, that's another option that I could be getting ready for. Okay, so then here's an example. And we went through these on Monday and set the context for them. So I removed those slides and I just have these example questions. And the wording might change, but that's a good question for our exam. And again, they were on our recording on Monday, and according for today, so you don't have to scramble to write them down, but read through them, because we're going to look through them again and check to see if people have questions about any of these proposed questions, okay? And so right now, I think we're dealing with 8 questions, of which you have to write 5 for. It's nice to come up with another example, so if you have a question that you think is a good one, you should send it to me, because if it could be another option, well, that lightens your load for other answers that you have to put on the final, right? So be thinking about that. And does anybody have any follow-up about this question that they want to talk about today? And I'll read it myself. Some medicines classified as endocrine disruptors can be used on patients because they aren't dangerous at every age. Describe one or more drugs that are in this category. Explain the molecular cellular problems they cause, and explain why they can still be used as a drug for some patients. So we talked about examples of that. We have several examples of that we could use, and you should strategically say, yeah, okay, these are questions that we talked about in our course. They're not new topics. And in some of these cases like this one, I said, oh, that would be a good question for the exam. It's just a really nice thing. And we talked about examples, and so we talked about the idea of examples last time, and Bryce asked about that. So there's this idea, of not having just a superficial answer, and also not having an answer that immediately dives into details. We want to have a balance of using this example to explain the context and the principle and terms and how they would be relevant for other examples. And in some cases in doing that, you'd want to use more than one example. So maybe you've learned pretty good explanation from this example, but you want to bring in another example to especially highlight another feature. That's great. It's not good to have no examples, and it's not good to just have one example where you don't explain how there would be other examples. Even if you don't describe the example, say this would be common, this is a common thing. There are other examples of this as well. There are several drugs that we learned about. So it's good to have that balance. But you can drive into that and prepare that, and see that, something like this is going to be an option for our final exam, so you can start preparing it. It's a little different than our midterm, because it's not, it's not like that's our exam and that's it, but rather there'll be options, and if you see an option, you can start studying and preparing to write about that topic. And you might have to learn some more, review, learn some more, build a concept map of how you want to approach it, like that. I'm not going to say, oh, I showed those in class and none of them are on the final, right? I could change the wording around a little bit, but not to the degree that your preparation would be irrelevant, right? Yeah, Bryce, you have something?
Talking about the using multiple examples for a question like this, the one that we spent the most time with was Accutane, right? For this example.
Yeah.
And then one that you highlighted last class would be the valproic acid. How would you obviously you talk about Accutane and the sensitive period that goes on with that, but then how would you would a way to use multiple examples, and that would be also sensitive periods are seen in stuff like valproic acid. How would you use 2 examples to
Well, so let's say that you decided, I think, right, you're the one who brought up valproic acid. So you're like, I'm going to write about that because I already learned about it, right? I learned more than the classmates did because you read about it for Monday's class meeting, right? So you're going to go with valproic acid. But then you realize, well, when you start diving into the cellular molecular pathway, maybe valproic acid has a cellular pathway that is not very well understood. So it's a great example, except, dang it, that Accutane has what looks like a really appealing cellular mechanism, because the way it works as a medicine is it triggers apoptosis in those oil-producing cells. So that's just a sweet one, right? Because you're like, that's how that works for acne, you know? The patient is producing too much oil and they get bacteria growing in their skin because there's too many nutrients around. So we're just gonna kill some of those. And then we had this thing where we're like, whoa, and it's thought that that's also the way it disrupts fetal development, is it causes, it triggers apoptosis in a couple different tissue types. So that would be an example of that, where you say, you know, people are still learning about valproic acid, you know, they have a really good handle on like what it starts to cause, but you know, they're learning more about the overall pathway. Meanwhile, other examples are very well, you know, relatively well characterized, and so Accutane is one. I want to add some of that to there, like that. Yeah, and you know, you can. It's very, nobody could criticize using a couple of examples. That's actually great and preferred. You know, to get a good answer is use a couple of examples. What I want to avoid is somebody just saying, well, here's a list. That's it. You know, here's a list of examples. There's a lot of examples.
So
and limit it, to that. And say, well, I wrote a pretty long answer, and I'd be, yeah, you did, but it was just superficial because you just, kind of touched on kind of vaguely some examples. But 2 examples would be, great, like that. Yep.
Yeah. So talking about like what you said, like superficial answer, like would it be like more? Would it be better to like round out like an example like Accutane and just say like, oh yeah, I can like kind of pull all this together, or if like let's say valproic acid's really like a good example because it is a good example for a sensitive period, really, especially to be like, oh, talk about Accutane because it's got a very clear molecular like pathway, knock that part out, and then like kind of switch gears to Accutane or to valproic acid, or is it more important just to get a rounded answer? I understand this one whole topic very clearly.
Well, I'd say, part of this is like the kind of the logic of demonstrating understanding. Yeah. And part of that includes picking examples that are good, that's a good example, not just that you knew about it, but also, it fits in, like that. And that's one way we're kind of trying to set this up is, we should try to write about sensitive period, on more than one answer, because that's a big concept for disruption, is there's really like to be a sensitive period like that. So it's part of it is just picking a good example like that. But then, so I would say kind of, construct, because it, likely you'll find some gaps in your understanding of Accutane. So you would be like, I gotta get a little better handle on that, and preparing that as an example, definitely like that. And then, it's totally appropriate to say, if you got a good example you're gonna drive into, it's very appropriate to say, well, here's some examples of our course, valproic acid, and flutamide, and Accutane, just list examples that fit into this, that's great. And then if there's a feature, a detail of one of those to say, this is especially good example, like you just said, valproic acid is a very good example of sensitive period. You're like, oh my goodness, neural tube defects, oh my goodness, right? Because it's neural tube, for a very specific window. So, that would be great.
Like support one topic.
Yep, yeah, absolutely, yep. So there's an example of that. Other questions, comments, issues about writing about that idea of medications and the idea that some could be used even though there's a sensitive period when they're very dangerous. That's helpful. I can tell you about a noise that was happening like that last semester, and I just laughed about it and told the students about a children's book I read where that happened and kept teaching. And later, everyone was, in a semester, everyone was banned from teaching in that classroom. I was like, oh, I guess it wasn't. I guess the students were concerned. Yeah, all right. So other issues of this question, this topic. And, you know, you can see, you know, what, and we talked about it, right? It's a good one for our exam, okay? So then I said, this is a good one, just because it's just such a good setup. So instead of writing a more broader one and saying, well, what examples? This is, I think, the only one where I'm like, you could prepare an example on that one and just smash through really great things about this. You know, ranging, right, from, like, Ashton and I really like to go with molecular stuff and say the rest of it is, you know, a waste of time and energy. But Ashton wants this course to be done. But in fact, it's a capstone course for a good reason. It's got just ranging relevance like that. And so DES is great, topical, you know, and it's great to appreciate that range and cover it, you know, range across it and not stick with the fact that on my PhD thesis committee, one of my thesis committee members was a DES daughter. And she got pregnant twice while I was in graduate school, and we had our committee meetings in her home, in her bedroom, because the doctors put her on bed rest for the duration of both pregnancies, right? If I just wrote that for my answer, that's not a good answer, you know? I mean, it deals with the personal and topical relevance, but it doesn't dive in. And this is just a great one, you know, for diving in all the way to the molecular mechanism, and oh my gosh, Hox genes are involved, and we love that, right? Because, you know, those of you who took developmental biology know, oh man, Hox genes are just a home run. They were the start of the field of modern developmental biology back in the mid-1980s. Yeah? Okay, so issues or comments or questions about that one? Yep.
Is there a lot more to the source of DES other than it was a drug, like the backstory was a drug that was given for?
Yeah, just pregnancy. I'm pretty sure it's about morning sickness and mom's comfort during pregnancy. And so given to about 1 million pregnant women in the United States back in the late 1950s.
So just throw that out there and then do the whole molecular mechanism, the pathway of the.
Yeah, yeah. Julia, did you have something or no? Okay. Yeah.
And then the first part of the question is just the statement, right? There's no, we don't have to address the first sentence, basically.
Well, you would want to describe the reality, the situation of this is a drug that was given a long time ago.
Yeah.
And so to the people who got the drug, there was no impact because there was a sensitive period, right? So, you know, bring up sensitive period and all these other concepts, bring them up every time they come up, right? Each question and answer is really independent, right? Say it's another situation where it's a sensitive period, mom's not going to be impacted. Side note, right, we said, well, people don't use DS because it causes cancer at a low rate more generally, so it's you know, not a good drug, and they haven't found a use for it. So it's an example of a drug where it was used and it's no longer used, different than valproic acid, Accutane, so forth like that. And but it's good to write about that, right? That it's an example where it's a natural population, natural event, even though it was a prescription medication, you know, and it was in the past, right? And so you can bring up the idea of delayed response, you know, so sensitive period, but not considered, I would say not considered a teratogen, because teratogens cause kind of immediate observable birth defects. It's one of these delayed responses. You know, there are other questions where you want to write about the idea of a delayed response, but this is an example of that, and a really dramatic example of it, because the delayed response takes till the fetus gets to childbearing age, right? So 20, 30 years later, like that. So just a great example to set the stage. Not in a one sentence way of like that, but rather describe that. Because of that, you know, in our second half of our course, I think there was quite a bit of that broader context, sort of the societal context of these issues of pollution and so forth like that. Yep. Yeah, and we want to write about that. You should be writing about your capstones also, you know, about where does this fit in, and you know, how's it, you know, yep. Yeah, so although it is a statement, it's it should be expanded upon in an answer. Yep. Okay, so that's one we talked about on Monday some too. Yeah, great. And then this matchup. So you're welcome to write about your capstone. Your capstone example is great. And so here's an option to write about it. And hopefully, you know, write about it for that answer and not all your answers, to be like, oh, well, as it turns out, my capstone project was DES, so I'm going to answer it again, you know, basically photocopy my answer for 2 or 3 questions. Try to look at separately like that. I should mention that for those of you who had my classes before, I emphasize that when I grade exams, I grade 1 question at a time. And this is just a different exam. So I'm going to read all your exam and say, wow, you got a lot out of this course. You know, grade your exam. Which is, you know, a typical way instructors grade exams. It turns out that's just highly biased and inappropriate to grade that way. To just be like, okay, I'm going to read this whole exam. Because the student will have a good answer for the first question, and then you just breeze through it going, oh, that's a pretty good answer, you know. It's shorter, but it's still a good answer, right? So, but, you know, realistically, right, I got to really look at your exam. Yep. So have that in mind and bring different stuff to different places. You know, striving to do that, like that. Okay. So this one. Any questions, comments about that one?
Can we pick a course or an example from outside the course that is similar to our, but as long as it still fits into
Sure.
the idea of the course?
Yeah. And in your example that you want to line up with your capstone, you want to be able to elaborate on that example and not just be like, I know people smoke. Here's my capstone project, you know, right? So yes, you can bring an example, and in studying, that's what you're doing. You know, in this is you're trying to prepare, fill in gaps, like that. It's different than, you know, an exam where it's going to be like, oh, there's going to be a lot of fill-in-the-blank questions, you know, where you're trying to cover all the topics. You're trying to narrow in and prepare essay answers that fit into that. Yeah.
Would it be appropriate to, like, let's say there's an example from our course that kind of follows like a similar, like, affects the same like pathway as our capstone project, but it's from this second half of the course that it, like,
Sure.
you know what I mean?
Yeah.
That'd be appropriate, even though that it's one of them's a
Yeah.
graduate and one of them's
Yeah, that would be very interesting, right? Yeah. And, you know, remember our course is all about kind of these 2 parallel events happening. One is your capstone project, and one is our exams and what we've been doing in class. And they're really synergistic and overlapping. And so we want to approach this similar to how you've been approaching your capstone, and, you know, diving into stuff and learning stuff in detail. And the concept is, well, it's a core requirement, there are core requirements to take, you know, this course, and one would be writing intensive, right? So this is a WRI3 course. So part of this is the idea of, can you use your advanced knowledge in a particular field of study to be able to write well and pull things together? So, you know, that's why it's structured the way it is, right? I don't structure my other courses this way. Yep. Okay, so there's another one. And so this one is, I really said there are 2 sentences to it, and I wanted to put that second sentence first. So, you know, what is traditional toxicology? We learned some terms and concepts, but we really talked about it briefly, and instead we mostly emphasized the modern challenges that people faced, especially with these endocrine disruptors with regard to how toxicology had been functioning. They loved dose response curves. They loved testing a drug on one organism at one age. You know, we've got 1-year-old mice, we're gonna give them this new cosmetic, see if it bothers them. If it doesn't, we'll sell it, you know, like that. And so that's what you want to set up there, is this challenge that is brought to toxicology. And it's not a toxicology course, but it's toxicology that we've been talking about, and it is a challenge. So that's what this question is about, is trying to match those topics up and the way we cover them. Yep. And like I said, I deleted the slides for Monday, but on Monday I had some slides, and this one was a slide where it just lists features of endocrine receptors make it hard to study, and all of them are just playing off of what was a weakness in traditional toxicology that endocrine disruptors exposed. Weakness, traditional toxicology, we're going to expect a linear dose response curve like that. And whoops, not happening, you know, with endocrine disruptors, kind of the opposite, you know, like that. And just on and on. There were like 10 topics on that slide. So again, we had that on Monday, and that's what this question is about. And so it's a good question, I think, really good question. You know, these questions are supposed to be, you know, guiding you, but also opening up for you to get, you know, going on it. Yep.
So for this question, in terms of just trying to make it like not, I feel like this is kind of a difficult one for like the mile deep or the inch deep or the inch wide kind of thing.
Yep.
So would a good way to address it be like give some of the like you said, traditional toxicology, like the straight curve, and then they don't really think about necessarily a sensitive period or the fact, and then give like an example of like an example that we learned that addresses that. You know what I mean? That is why traditional toxicology has issues, like just straight list and then one example for each one.
Yeah. Different than I think all the other questions.
Yeah.
I think this question, it says, give as many examples as you can. And so, as a student, strategically, I would just say, in this answer, I'm just going to give numbered examples and see how many numbers I can make. And for each number, I'm going to write 3 or 4 sentences. Here's an example: dose response curves. Blah, 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 blah. And then you shift gears to sensitive period. And then you shift gear to species specificity, like that. And you know, you just say 1, 2, 3, 4, 5, 6, 7, 8, and I go like, oh wow, look, you know, Juliet had 9 examples. I can see them already, you know, and that's a good bunch, you know. That's how I really dive into this one. The other ones are more essay, where it's one example and you try to dive into it, and I don't really think that would be structured to have it numbered. But this one, I think I would just number them and say, I'm going to talk about this example now. And if you don't number them, they could be short paragraphs. I'm going to this one, now I'm moving on to sensitive period, you know, now I'm moving on to allelic variation in the population, so on, like that. Yep. And just see how many you can produce, like that. Yep. And because there's quite a few. Yep. Okay. Okay, so that's one. And then writing about the neuroendocrine axis in both parts of our course, right? And we talked about that some in our course too, about how, gosh, you know, the endocrine system, the neuroendocrine system came up a lot in our course in both halves for overlapping reasons. So write about that, give examples about that from throughout our course. Because it was just and you know, we don't need to restart our course and say, okay, lecture 1, but it just comes up a lot. Yeah. And you know, it's interesting because the neuroendocrine axis is a big part of development, but it's not like it's just, oh man, all we talked about it. It's like half of our, you know, development is about that. It's not. But in our course, probably about half of all the course on both halves of it is about, well, it's about hormones, how you get messed up with hormones, or how you can adjust hormones and do all kinds of stuff to development. Yeah, yeah. So that's, it's just a big take-home message for our course, is that reality. Yep. Okay, and then here's one, where I said, let's focus in on this idea, and we added to some of that information on Monday, pick one of those to explain the principles here, and hopefully, a different one. You can use the example more than once, but I just want to avoid you coming in and going like, yep. Because that's what I would have done in college. If there was an angle, I'd just be like, yeah, well, here's my exam. And it's the same answer, and I just put 1, 2, 3, 4, 5 in front of it. What are you going to do about that? And we're just like, oh, that was not my intention. So you don't have to feel like, oh man, I cannot use the same example again. But just strive to bring in a bunch of learning. And because we have so many examples that can illustrate it. Here, it's a really kind of emphasized one, and we had some good examples, and we added a little bit to that on Monday. Yeah, and Mike brought this up, but yeah, if you have one that you were like, oh my gosh, that's from one of the Monday discussions that we didn't even talk about in class because you're shy, or you weren't here. Go for it. We are not restricted to learning that we did, from our reading or our class meetings, or your capstone, of course, But yeah, and again, we're hoping to talk about some evolution stuff today, and there's this optional reading. So, if you decide, oh, I'm going to write about that question, it would be a good idea for you to page through the reading, learn a little bit more about it, write that, preparing. So I expect that also, not for all your other exams, right? You're just like, I got to study, I got to look back at everything we did. For this exam, you're targeting it and you're filling in gaps, but you're also, those gaps may be new stuff, and which is perfectly great. To bring in, oh, this is a different example, but it's really cool. And, that's the way to study for this, is to not say, I'm just limiting myself to just trying to grind through it, Because a lot of stuff we covered superficially, and we should learn more about it, because we talked about valproic acid in class, like you looked at it some, but the other students didn't have heard of it like that, or that, yeah, other examples. Yep. Okay, so that's one like that. And then we should write about forever chemicals, and that goes to this idea of topical, right? That's part of the capstone is, oh my gosh, this is particularly relevant. And one way to think about that is, every once in a while, these people who run the core check in and say, well, what's it like? And I'm like, oh my gosh, we're writing about forever chemicals. And they're like, ooh, literally, ooh. That's something, right? But not just at the level of the core, in the level of in the context of the bio major. We can really dive into forever chemicals and understand those mechanisms. Like, it's very, very common that they're endocrine disruptors, and that's a major issue with them. Well, that's not on the skillet that you buy at the store, right? It just says no forever chemicals. But it doesn't say anything about, the biological impact of the forever chemicals like that. And we talked about some, and like there'd be good examples where we talked about somewhat superficially, right? I brought in dioxin. Dioxin's a great forever chemical because I'm Dave Oxen, right? I burn trash in my backyard every week as a kid, and I just must be 50% dioxin. And running along, right? But I bet when they try to cremate me, they'll be like, he can't burn. It's still Dave, right? It'll be like, I guess it's just forever chemicals, pretty much from head to toe, right? Like that, So, dioxin is one I'm interested in, and maybe you can run into one where you want to fill in some more, so you can write about it, And don't just read about it, to take notes and organize your notes so you can kind of craft the answer like that. So questions or ideas about this one. That's a good one, just to force you to make sure you get some topical writing if you choose to write about this one, in the context, but then dive into it. Yep, great. And then, and here's this Evo-Devo question, and we can talk about that more today. And I'll put some optional reading. If you don't have to do all that reading, you could look at it just in a strategic way to try to fill in gaps and stuff like that. So that's one. And then, like I said, if anyone suggests one, it's possible I might decide I see a gap, because I'm thinking about this too. But we could have, write 5 answers about 8 questions or 9 questions. Maybe there'll be 9 or 10 questions. But you'll hear about any other examples soon. And again, some of the language might change a little bit, but it's not going to be a thing where you want to slash the tires on my car, like I used to do to all my instructors. One time I said in class, I used the example in class of slashing the tires on my car, and I said, I have a red Corolla. And a student raised her hand and she says, I have a red Corolla. And I said, okay, calm down, let's not slash red Corolla tires. So, but yeah, that's that. Okay, so that's where we're at on that. So what I have next is more slides about that, talking more in a new topic kind of way about this topic, setting it, driving into that topic some about how this fits into evolution. And we'll go there, but how are people doing? Do people have issues of other questions or anything like that that they're wondering about and thinking about? Yeah, Mike?
Is the test gonna be like the same length as the first test, or should we plan on it will take us the whole 2 hours to write out the answers?
Oh, well, yeah, because you have to plan your social time. Yeah, I always have tests that are intended to be during the regular class meeting. Just because that's what I'm always anxious not to craft an exam where students are like, I did not have enough time, because shouldn't be part of it. So we're scheduled for 2 hours, and 2 hours should be plenty.
So in that case, like, let's say we did 5 questions in an hour and 15 minutes.
Yeah.
We have 45 minutes left over. Can we do more questions for extra credit? Or
no, we should focus on 5, planning to answer 5, and yeah, that would and also because then it brings up the idea of I answered 7 kind of okay, answer 5 well and be done. Yep, yeah. And just because you reminded me of an anecdote from way back, a student named Chris, who's a genius, used to take exams, and he was in my research lab, and he'd be back in 10 minutes. And his strategy was he'd start taking the exam, he'd get some answers down, and he'd start thinking whether it'd be more beneficial to try to answer more questions or to come up and just slam the test down and walk out and actually lower the grade and the score average by just bumming out the classmates, because they're just like, God, that guy's done already, and I'm still just struggling, and I don't feel very good about myself. Yeah, my gosh, yeah. So that's what I thought you were driving at. It was like, if I can get done in 10 minutes, is that a good plan? Oh my gosh, yeah. You'd just be like, wow, I had not thought of that strategy. But he had that kind of charisma, because he would just, I don't want to do it because it'll sound like a gun job, but he'd just go like that. People are just like, I'm heading my exam in now too. And it would be 10 minutes. He would just answer like 8 questions. Be like, yep, crushed it, and get like a C-. So anyway, okay, good. So that's a good question though. Yeah, 5, focus on 5. Yep. Other issues and stuff like that. Okay. And then, our exam's gonna be a Wednesday, so a week from today. And my intention is to give you whatever resources, you know, you'll need in the next 2 days, right? So I'm not gonna send out another question on Monday or Tuesday. So if you wanna think of a question, suggest a question, or a change to one of these questions, let me know, you know, in the next 2 days. And I'll let you know of either, you know, an additional question from me or a kind of a twist on a question, or, you know, from a student, yeah. And I super welcome that, you know, because you're thinking about this too, and, you know, when I was in college, I thought about that too, and I'd be like, you know, we learned a lot about this topic, and it's not represented yet. I wish it would be, you know, because I could write a good answer. So I'm always open to that. Yep.
Will you just like give us all whatever 8 or however many there end up being questions and just say pick 5, or will we have to pick from some of them? You know what I mean? Like, will we, like, there are a couple topics that you cover, like, there's some that connect to the first half of our course, will we have to answer one of those, and then like?
No, it'll just be, yeah, like that. So, yeah, so if there's one of these questions and you're like, swoosh, I don't like that question, I wanna avoid that question, you just have to make sure you pick 5 others. But you're not gonna be, and it's not like, and another thing to say is not like any of the questions will be worth more than others. Remember, on our midterm, some are worth twice as much as others, right? They'll all be 5, it'll be, you know, pick of these, they'll each be a 20-point question for 100 points on the exam. Yep. Yeah, and then also, if you have questions, you know, let me know. We can meet, again, not just about the capstone, we can meet about that. But we're gonna meet about the exam and talk through stuff, talk through your ideas and stuff, get clarification. Because of that, also keep your eyes open through, you know, next Tuesday, because sometimes I'll be like, oh my gosh, I gotta notify everybody. So I'll post something on Brightspace and I'll say, oh, you know, this is important for everybody to see, right? Just a clarification like that. So, you know, just be aware of that there might be a Brightspace announcement, just as a, you know, something came out and, you know, I wanna, you know, add a feature or something like that, you know, just a little, you know, not a big thing, but something. So just be kind of aware of your Brightspace. And my understanding is other instructors will do that too. They'll be like, oh my gosh, you know, I've completely made an error, you know, and I need to notify everybody. So you should be aware of Brightspace announcements right up till, you know, the day before the exam. Yep. Yep, okay. All right, and so we can still keep talking about that, but, you know, if I could talk more about this evolution stuff, I will. Okay, so here's some evolution stuff, okay? Oh, this one is a little bit out of room. Yeah, out of place. So, okay, so there are 2 situations we should be considering with regard to this topic. And so the first one is examples when a species has phenotypic plasticity and it is lost during evolution. And the other is examples where a species does not have phenotypic plasticity and it's gained during evolution. And both those are considered to be a big topic in evolutionary biology. Pretty simple. Either it's lost or it's gained. Yep.
In the first set of workers, did we talk about the second one?
We can talk some about the second one. But just because the first one is the one we've dealt with in our class, right? Because we have a couple examples. So I pulled those up. And so, and again, you know, talking about new material, but also talking about that question. And what's that question about? Because we haven't had a chance to talk about it much, but we have talked about it some. So that first one is it's understanding that there was phenotypic plasticity and had been lost. So what's that like? And the answer would be closely related species show phenotypic plasticity, this species does not, okay? And we have 2 great examples that student brought up in class. Bryce brought up one and Emily brought up one. And always remember that the phenotypic plasticity could be a polyphenism or a reaction norm. It's just always easier to go to examples of polyphenisms, one or the other, but it can be either of those. And so here's one that Emily brought to a class discussion. So there's this fish that lives in, I think, Death Valley. So it lives in a desert in a little tiny pond, and it has close relatives that live elsewhere. And this would be an example where it appears that this fish has lost phenotypic plasticity, and it has to do with regulation of development by thyroid hormone. So it's a good example for our course about the regulation that's occurring. And the regulation occurring is normally found in other closely related fish species living in other places. And in those other places, those species see variable environments. And so they have phenotypic plasticity on details of how the thyroid hormone is regulating their development. And it would be good if you were going to write about this question to pick an example of this and explain it. So, look into it, right? Go back and look at Emily's example and learn about it a little bit so you can explain that concept. That this one seems to have lost it. And it seems to have lost it because it lives in an extreme environment. So it doesn't see variable environments. It sees a pretty constant environment, and it's out at an extreme of what the other closely related species experience. But this species doesn't vary it, and it's been living there for a while. It says, I think, on the order of 10 or 20,000 years, like that. And during that process, it seems to have lost the plasticity, which is not surprising, but an example of evolutionary change. Yeah?
Which on the previous slide, what is this put under? Or yeah, there. Polyphenisms, or sorry, the next one.
Oh. Yeah, I would say reaction norm. Yep.
Can you explain that a little bit?
Yeah, so that's where, just to provide comparisons, that's where there's a graded phenotype depending on environmental settings. So that's in contrast with a polyphenism, which is like colorful butterfly wings or plain butterfly wings, one or the other, versus this gradation like that. Yep.
So yeah, pretty much all what we're talking about in all of these is going to be reaction norms.
Oh boy.
This question.
Oh yes, I'll say yeah. I mean that's kind of a broad generalization, so I
Yeah.
don't know, but
Wasn't the toad example one of them?
Yeah, that one's also
Yeah.
a reaction
Oh,
norm.
it was?
that's a reaction norm also, yeah, because it's the timing of their development.
Oh.
Yeah.
Okay.
Yeah, and I have that on here too. But yeah, I mean, I'd say it's good to know what the example is that the examples that they're going to be writing about,
Yeah.
what is going on, so be able to name that. That's why I brought it up, is because what is this we're talking about? And then, yeah, I mean, a lot of times I have to think for a while to think about whether, would those be some examples of polyphenisms?
You could do
Well,
like
so
fish or something.
I would say, like Luke, right, had the aphids that were winged or wingless, but was there whoosh? Because there would be other, I don't know about that. That might be an example where if the other, like closely related species of aphids are always winged to females, and then this, no, I guess that isn't lost though. Sorry. Yeah, I don't know. Yeah.
I was gonna say there are, I can't remember the species name, but there's a species of lungfish that is, it has gills and lungs, but then some of them are born with a ballast tank and some of them are born without the ballast tank. So that's a polyphemism that developed in the last 50,000 years.
Yeah.
But I can't remember the species name, but you can just look up lungfish.
Yeah.
That's a good one.
Yeah.
But there aren't many, like you
Right.
just hinted
Yeah.
at that. Yeah.
Yeah. Yeah. I mean, we could probably phrase a question for AI to address that, but right now, yeah. I'd say this pupfish is a reaction norm, looks like a reaction norm. And And so that's the article that Emily had found and brought to class, and plasticity first, phenotypic divergence. And so we all got that link. If you were going to write about this question, you might go back to that example, or Bryce had an example. This, I think, is a really good example because just in the abstract, they do a nice job writing about the concept like that. And because that's why they're studying the example in their case. They didn't run into it. They went looking for a good example. And and so again, some of you have already decided you're not going to write about this, so I don't want to belabor it too much, but I'm trying to guide guidance for everybody to learn some about this. And that's that example, okay? And then Bryce brought up this example of some frogs, and that example is closely related species had this phenotypic plasticity, and then this particular species had lost it. And a couple of terms came up that would be relevant for Emily's topic too, but they came up when we discussed them in class, and one was this evolutionary term, genetic accommodation, and then the other is this idea of canalization. And so I just wanted to bring up that canalization again. We talked about it some. So this is what 1940s, a geneticist drew this map of the movement from the organism from genotype to phenotype. And the surface is, it gets plugged in different ways, but the surface for us would be the environment. And so the environment leads to different phenotypes. And so here, the surface are valleys, grooves, or canals, and there are multiple shallow canals. For the spadefoot toad or the pupfish, the organisms become highly canalized. So that's a term that means no phenotypic plasticity. We're going to go down development rigidly and robustly to a specific phenotype. And so that would be an example where, terms you want to dive into to, describe in the context of evolution and evolutionary change. Great. Okay. So just, to talk about all of us about what's going on with this topic. Okay. So genetic accommodation, we used both the terms genetic accommodation and assimilation, and we talked about them that day in class. And it's good to look back at those terms. It's good to look at all these things because you might realize that some of this has relevance for another answer, that you want to write about. Yep. Okay.
What's I'm looking at those definitions of accommodation and assimilation.
Yep. So assimilation is like a subset of accommodation.
Oh, okay.
And
What do you like?
well, so I would say that the examples we just talked about are accommodation. I think, well, I have to think about it. Maybe there are examples of assimilation, because the idea is assimilation is it gets so canalized that you don't even need to be living in a desert to have the pupfish develop these traits. Yeah. Okay, just some of that, okay?
Is assimilation like
Oh.
the extreme? It's like the when it,
I would say, yeah, like I would say that that's when like the evolutionary change is being locked in. Yep. I think this slide might be out of place. No, it's not. It's just the idea of the concept of the phenotypic plasticity creates variation in a population, and natural selection works on variations in a population so that it can be of a selective advantage. How this is out of place is just, it's more about that second example of evolving phenotypic plasticity rather than losing it, okay? And here's the second example that we haven't run into examples of. So it's examples when a species does not have phenotypic plasticity and it's gained during evolution. We've talked about it a little bit, but pretty briefly. So we should talk some more about that, because that's...
Just everyone that has phenotypic plasticity.
What?
Wouldn't that just be true of all phenotypic plasticity?
Oh, that it didn't have it and it gained it?
Yeah.
Yeah, so I think that's an interesting question. And the way I would express that is when, over an evolutionary time, there will be an ancestral condition and then a derived condition, like that. And the ancestral condition is really and derived condition are really temporal. This was the old one, and this is the more recent one. So, which comes first? Trait or the phenotypic plasticity for a trait? Yeah, and in our examples, in our course, those examples tended to be bold, kind of dramatic examples, like winged and wingless aphids. And in that situation, in our examples in our course, we would say, yeah, there was the ancestral trait was being able to make wings. And then the derived trait is whether you're going to make them or not. That's the phenotypic plasticity, like that. And so I think for our course, it would lead really toward your thinking. I'm not an evolutionary biologist, so I have a hard time thinking about theoretical, evolutionary topics like that. Would there be first phenotypic plasticity, and that would be the ancestral trait. But I'll try for an example. And so this is maybe, coming up with an example is not always just like, oh, that's great. But if you think about it with regard to regulating body temperature, the ancestral trait was not regulating body temperature. So there would be phenotypic plasticity. And that's an odd trait to think about, phenotypic plasticity, but like a frog shows phenotypic plasticity with regard to temperature because its body temperature is controlled by the environment. And a phenotype would just be activity, where it's activity dependent on temperature and it's just directly proportional. So that would be an ancestral characteristic trait, and then the more derived trait would be homeothermic. Is poikilothermic like cold-blooded? I don't know the terms, but like you start out with cold-blooded organisms and then you evolve warm-blooded or organisms that can control their body temperature, and so those would have lost their or not have phenotypic plasticity. But I'm just kind of winging it with trying to think of that. But I think when you start thinking about evolution, I would say, yeah, maybe that is, maybe that the ancestral being plasticity, and then the derived trait being more lack of plasticity. But I think for our course, most of the things we talked about would be, yeah, obviously they had to have the trait first and then they evolved the plasticity around the trait. Yep. Okay, so, but that's what we're talking about now. This examples when phenotypic plasticity is gained during evolution, okay? And so in that context, then the evolutionary biologist says, how could it be gained? You have a trait, and how can you gain this plasticity? What's the evolutionary mechanism? So that's what we want to talk about really for the remaining time we have, which is just a short time. But how do you gain this plasticity? And So there are these two, I'd say, schools of thought about evolutionary change, and one is that evolutionary change of any kind involves small-scale changes in multiple genes. And the other is maybe there could be some major changes in a few genes, like that. And in developmental biology, developmental biologists have found, there's lots of regulatory genes that control big picture items. Like I mentioned last time, the idea of an elephant with muscles in its trunk. Well, we have muscle genes in our nose, and an elephant's trunk is a mammalian nose. So in that sense, maybe evolution of the elephant trunk involved just activating a lot of pre-existing modular type pathways where you're gonna need some muscles in that to move it around. And you could activate by mutation, right, like that over evolutionary time, activate expression of the muscle genes and producing muscle tissue in nose. And because of how developmental biology emerged in the last 50 years of the field, there's lots of developmental biologists who imagine that the dominant form of evolution is just throwing some big switches versus little tiny changes, a whole bunch of maybe even sort of marginally relevant genes, like that, okay? And so that's the reading I have. The author doesn't even say that because the author is driving forward. And I brought this up last time, but I didn't really talk about it. Where the author writes this, Emerging Principles of Regulatory Evolution, of the optional reading, where the author really wants to talk about, look at how there's a variety of modular, big picture regulatory events that happen during evolution, and that's going to be the way these organisms evolved, like that, okay? And so that's what this second part is. The second part is, well, what is it like when phenotypic plasticity evolves, like that? And just to cut to a couple of examples, so when I talked about my insect, I said, well, this insect can diapause. It can overwinter, like that. And how does it do that? And when you look at insects, lots of different species, lots of closely related species, and you see isolated species can diapause, can overwinter, and others don't. So the interpretation is it's like very easy to evolve this developmental arrest. And different species evolved it to stop at different times. So like around here in the winter, you can go out and you can go to nature hike and you can find like little insect eggs under a leaf. Well, those insects evolved overwintering, diapause arrest, by stopping their embryonic development in the egg and just waiting. And they have to choose to stop it before freezing conditions arise. So somehow it's a decision they're making. It's a phenotypic plasticity. And along with it, it's anticipated, based on other research, they're extending their lifespan. They're also, activating FOXO to last through the winter, like that. And other species have arrested diapause at different stages. There are some we can actually find a little curled up caterpillar in the winter in Maine, just curled up like inside some bark, like under a tree bark like that, or a rolled up leaf. And that's where that species evolved to arrest over winter. My species, very common, in species is that it stops as the pupa. And that seems like, a great time to stop, like that, okay? And so, I don't have a slide of it, but the life cycle of an insect like a butterfly, moth, social insect, beetle is going to go, egg, embryo, then larva growing, then pupa, then adult. And the pupa is like a resting stage, hiding in a cocoon or under the ground, hiding somewhere. Seems like a great stage to overwinter. And so when you look at these species, you see that these different ones evolved the ability to stop their development in the pupa. And they're independent, you can see from the evolutionary tree. This one's evolved it, and there's one over here that's evolved it. So many, many examples of independent evolution of the ability to arrest during the pupal stage. So then, if you look at the pupal stage, it really looks constant from the organism, right? The organism is like a caterpillar, and it's going to shed its skin, and now it's going to be an almost entirely immobile pupa for a couple of weeks in the summer or over winter, like that, okay? So it looks like it's just, there's the pupa. And so when you look inside those species at the development of the animal inside the pupa, metamorphosis is happening. And in the first, we'll say, third of pupal cycle, portion of the life cycle, the pupa is like a stew, like a soup with little pieces in it, because it's just destroying the caterpillar almost entirely. Some caterpillar tissues are retained, like the brain and some tissues, some muscles and so forth like that, but most of them is just getting wiped out by autophagy and apoptosis, and we're going to build the adult from that. And so that would be a very dynamic period of development. And then if you look at the last third of the pupal stage, it just looks like the adult curled up. Looks like everything's done. And it is, largely done. And it's just waiting for the right time to emerge. And so if you saw what's happening inside the pupa and you said, okay, so this organism is going to evolve phenotypic plasticity, how's it going to do that? You would say, I bet it's pretty easy to arrest in the pupal stage at the end of it, because it's just a moth. It's not even developing, it's just hanging out, it seems like, right? But with the exception of only like 2 of, say, 50 or more species, 2 species do that, and the rest, like mine, all stop in the first third, when it looks just like a soup, and there's just crazy changes happening. And it doesn't stop because it's cold, and anticipates that by short day length in the fall. It says, this is when my species stops. If we detect short day length as a caterpillar, we're going to get to the soup stage, and we're going to pause there. So you just that's just ridiculous that all these species found that was a convenient or appropriate way. It looks like the answer is, based on my studies in my species, is my species stops because it stopped making the ecdystrophin. And when I first read that, I was surprised, because that's the molting hormone, and it functions like a ballistic. It goes boom, molt, boom, molt, boom, molt. But it turns out low levels of that same hormone are needed for metamorphosis to continue. So it's playing a tonic role, like a foot on the gas, just gently driving it forward. And if you remove it, everything stops. And so that just opens the door for the immediate interpretation of, this is a highly modular situation where you can have one thing stop and create the phenotypic plasticity. All you have to do is make mutations in the regulation of production of that hormone, and you'll have arrest. It'll be in that soup stage, but turns out, although it doesn't look like a convenient stage, biologically, biochemically, it's a perfect stage, because that hormone is controlling all of the events, the death and the development, the growth and stuff like that. So that would be an example of this idea of evolving that evolutionary phenotypic plasticity. And would it be like, does it evolve because it's taking advantage of like a modular aspect of development that is like, like a sweet switch spot like that? And so that's relevant for our talking about phenotypic plasticity and evolution also, is the emergence of phenotypic plasticity and what can we understand from development that could help us appreciate that. And from development, that's where these people who really liked the idea of the big principles of development being relevant for the evolution of phenotypic plasticity, that's one place where they say, look at these examples. because we see examples where there are modular, specific regulators of big parts of evolution, or big parts of development, and if we just mutate that, alter that, adjust that, the whole show will change, like that. A good example of that is think about that and think about any of the examples we talk about, like those aphid wings, right? They make wings or not. And I don't remember the details, but I think that they don't make any hint of a wing. You know, not like a little nub, you know, or a little crinkly thing, but it's just like that. So, you know, the hint there is that phenotypic plasticity evolved by some developmental mechanism that includes a feature that allows you to just say, do not make wings. And the ancestral trait would be the ability to make wings, and then you're saying the way that evolved, like happily, luckily, was a modular thing, switches like this, and it leaves us open to evolving phenotypic plasticity because we have these whole modulars, you know, like modules, like this module controls make a wing or not. And it sets it up for easy phenotypic plasticity. So that's what that's about, and so that's what that reading includes too. And it's you know, it's a little higher level pulling things together for our course, but some of you, there's a good chance you'll see that, and you know, it's got to be at the end of our course because we have to have a bunch of examples for you to think about, I could see how that evolved. And so I just, because we're running out of time, I have some slides about it, but you know, just that idea you can think about with my moth, where first you discover, oh, this hormone is running the show. And that would be a great time to arrest development for the winter, because all you have to do is make that hormone stop and the whole show stops like that, you know, even at normal temperatures. We do that in lab where we have one room that can be a short day room. The lights are on for just a short time. And even though everything else is the same, normal temperature, you know, and food and stuff, those animals will stop as soup and just wait. Just remarkable. So that's what this is about. And that's about this discussion of, does evolution occur? So does the setup, the ancestral setup, did that occur because we had modular evolutionary changes to set the stage for being able to make a wing? Or is the ability to make a wing leading, you know, up to the evolution of insects? Ancestrally, insects did not have any wings, and they evolved wings. So did that involve little tiny changes of a bunch of genes? Because if formation of a wing involves little tiny changes of a bunch of genes, well, it seems like that would be really hard to evolve a phenotypic plasticity of either having a wing or not. It seems like you wouldn't have a wing or not. It seems like you'd have kind of a crinkly weirdo thing that was maybe smaller and less energetically consuming to produce, but it wouldn't be no wing or wing. Yeah, so that's what that's about. And again, we've run out of time for our course, and that's, you know, that's fine, because this is, again, an advanced topic, but it's very interesting, and people are very enthusiastic about the topic we talked about in our course. The first half, this is all about her first half topics. Those topics are very, very interesting for evolutionary biologists and learning about them and stuff like that. And so I'll put that reading up. If you want to chase down that question some, right, you'd have to do some of that reading and look into it. Or, you know, again, you know, that's good reading for you to read, you know, after our course is over too. Because it's about evolutionary biology, which always requires some, you know, serious thinking and kind of sleeping on it and stuff like that. So that's where we're at. All right, great. So we should be all set up, and we'll roll forward towards seeing you again a week from today, when Mike is going to finish the exam in 10 minutes. Woohoo!